Good morning, everyone. Welcome back. I'm Steve.
And I'm Scott.
And together we're Backyard Musings, Apple Valley, People's Republic of California.
And we are talking about a health issue, science-related health issue that I think everybody's going to want to hear.
Yeah, and this is very hopeful. I like this.
It is.
The discovery of a defensive mechanism could help stop cancer before it spreads. Cancer cells rapidly increase their energy output when physically squeezed, according to a study in Nature Communications on July 30th, 2025. This immediate burst of energy is the first documented defensive response that helps cells repair DNA damage and endure the crowded conditions inside the human body. So if you got cancer inside, it's like multiplying, right? So it's like growing. So they're talking about squeezing it. I don't know how. The results help explain how cancer cells survive mechanical challenges such as crawling through a tumor microenvironment. That just does not sound right.
Does it not spread? It usually, I mean, not all of it maybe, but doesn't a lot of cancer spread through your blood?
I don't know, man. I'm not an expert. I don't know.
Because your blood goes everywhere, right? Blood vessels.
Well, this is what it's saying: slipping into porous blood vessels or withstanding the forces of the bloodstream. Identifying this mechanism could point to strategies that hold cancer cells in place before they spread. Would you like to know more?
Researchers at the Center for Genomic Regulation, CRG, in Barcelona, I'm going to assume that that's Spain, right, uncovered the effect using a specialized microscope that compresses living cells to about 3 microns in width, roughly 1/13 the diameter of a human hair.
1/30.
Oh, 1/30. Oh, that's even smaller. That's pretty tiny.
Yeah.
They observed that within seconds of compression, mitochondria in HeLa cells, HeLa cells, moved to the surface of the nucleus and delivered extra ATP, the molecular energy source used by cells. Quote, it forces us to rethink the role of mitochondria in the human body. They aren't these static batteries powering cells, but more like agile first responders that can be summoned in emergency situations when cells are literally pressed to the limit. This is according to Dr. Sarah Delcy, corresponding author of the study.
Very interesting. The mitochondria formed a halo so tight that the nucleus dimpled inward. The phenomenon was observed in 84% of confined HeLa cancer cells. And like Scott said, it's spelled HeLa, and the H and the L are capital. The E and the A are small, compared with virtually none in floating uncompressed cells. The researchers refer to the structures NAMs for nucleus-associated mitochondria. To find out what NAMs did, the researchers deployed a fluorescent sensor that lights up when ATP enters the nucleus. The signal soared by around 60% within 3 seconds of the cells being squeezed. Quote, it's a clear sign the cells are adapting to the strain and rewiring their metabolism, says Dr. Fabio Pezzano, co-first author of the study.
Okay, subsequent experiments revealed why the power surge matters. Mechanical squeezing puts DNA under stress, snapping strands, entangling the human genome. Cells rely on ATP-hungry repair crews to loosen DNA and reach broken sites to mend the damage. Squeeze cells that receive the extra boost of ATP repair DNA within hours, while those without stopped dividing properly. To confirm relevance for the disease, the researchers also examined breast tumor biopsies from 17 patients. The NAM halos appeared in 5.4% of nuclei at invasive tumor fronts versus 1.8% in dense tumor core, a threefold difference.
Yeah.
Quote, seeing the signature in patient biopsies convinced us of the relevance beyond the lab bench. According to Dr. Rito Brada, she goes by Rito. I'm going to assume maybe that's not a woman, or maybe it's a man. Rito Guse, co-author of the study.
The researchers were also able to study the cellular engineering, which makes the mitochondrial rush possible. I like how they're calling it mitochondrial rush. Actin filaments, the same protein cables that let muscles flex, compound around the nucleus, while the endoplasmic. I immediately thought of ectoplasmic from
endoplasmic reticulum.
Yeah, but I thought in my mind, I thought ectoplasmic from the Ghostbusters. While the endoplasmic reticulum throws a mesh-like net. Okay. The combined scaffold, the study shows, physically traps the NAMs in place, forming the halo-like structure. When the researchers treated cells with latronsulin A drug that dismantles actin, NAM formation collapsed and the ATP tide receded. If metastatic cells or metastatic cells depend on NAM-driven ATP surges, drugs that block the scaffold could make tumors less invasive without broadly poisoning mitochondria and sparing healthy tissues. Quote, mechanical stress responses are an underexplored vulnerability of cancer cells that can open new therapeutic avenues, says Dr. Verena Ruprecht, co-corresponding author of the study.
Okay, we got some co-authors on this thing.
Yeah.
While the study looked at cancer cells, the authors of the study stressed that the phenomena is likely a universal phenomenon in biology. Immune cells squeezing through lymph nodes, neurons extending branches, and embryonic cells during morphogenesis all experience similar physical forces. Wherever cells are under pressure, this is a quote, a nuclear energy boost is likely safeguarding the integrity of the genome, concludes Dr. Delsey. Going on with the quote, it's a completely new layer of regulation in cell biology, marking a fundamental shift in our understanding of how cells survive intense periods of physical stress, unquote.
So, I like it. This is great.
I like it. I don't understand a bit of it, but I like it. If it's trying to fight cancer, then that's the way.
You know how Trump is pushing for AI all across the board, and in particular to fight children's cancer? Maybe AI could be used here. I mean, let's get this nipped in the bud.
As long as it's good. Yeah. As long as the AI is good, because I know there's some bad AI out there.
Bad AI?
Bad AI. Yeah.
Bad robot, huh?
Yeah.
Okay. Thanks for joining us, folks. We'll see you in the afternoon.
Take care, everyone. Thanks for listening.